as Philippe mentioned, we have been for quite some years now studying how bacteria interact with the host tissue when it's present inside the organ. And it may seem weird that my location is Karolinska Institutet, but that we are at a location which is the Swedish Medical Nanoscience Center, which we were just about to establish. And there we use a lot of organic bioelectronic devices. We're developing devices to help us in these kind of studies. And I hope I will have a second at the end to show a few of those things that are relevant for the present work. But I'd like to first start from the ordinary infection routes. In the human, you have the epithelial linings that are continuously exposed throughout the lifetime to bacteria. Now, they were for a long time considered to be like physical barriers, but it is known today that they act as sensors with very sensitive systems to recognize the presence of bacteria in order to elicit the proper immune response. Now, studying these kind of mucosal interactions with the cells have, for quite some years now, been performed primarily in what we call a reductionistic approach, which is that you start off considering the intact organ where you have your infection normally, which has the presence of the, in the case of the kidney, we have the tubules, the vasculature, all the different cell types, effects of hormones and so on. And you kind of dissect out the system to eventually reach the cell culture phase, where you have a purified or an isolated cell culture that you use in your investigations. Now, this has for many, many years been really important technology, and there are lots of studies that have not been able to be performed without this kind of data. And also, I think the previous speakers today, some of them have also alluded to the very nice information you gain from these systems. And I'd like to emphasize that it's about 10 years ago that we got to see the first issue of the journal Cellular Microbiology, emphasizing the role of these kind of studies. Now, when we are considering the infection, though, if you think of a skin injury, so if you get a little splinter in your finger, that may be contaminated with bacteria. Now, as soon as they get into the tissue, immediately lots of signals will be elicited here, which eventually leads to the recruitment of immune cells to the site of infection, where they now will engulf and clear the infection. So, question is in our work, how can we study this kind of complex tissue response that is not possible to study in the cell culture dish? Because if we do so, we hope that we can bring this one step further now, taking the field from the cellular microbiology to what we call tissue microbiology. So to do so, we are using the reverse reductionistic approach, which is that we're utilizing the multiphoton microscopy in order to be able to study what happens in the intact organ. And I will not talk too much about the technology because that was presented earlier this morning. But I'd like to comment on that from the renal perspective. This work has really been pioneered by Professor Bruce Molitori's laboratory at Indiana University School of Medicine. So we're focusing on the urinary tract. Now, that's a great organ, we think, not only because we know the pathogen that caused the infection, we can do lots of genetic mutants and so on, but also from the tissue architecture. Because you have the filtering unit, that is the nephron. So you have the glomerulus where the blood comes in and is filtered, and the primary urine goes through the proximal tubules via loop of Henle, distal tubules into collecting duct and down to the bladder. Now, the kidney is extremely well vascularized. So that means that in close proximity to these epithelial linings, we have lots of peritubular capillaries. So in order to image this now in real time on the microscope, we're using a rat model where we now can exteriorize the kidney so that the kidney is still connected to all the blood flow and everything. We'll just put it on the outside of the skin. And I think this is a point that is worth emphasizing, that these kind of studies actually are performed in the live animal and not in an explant, or that is an explant you can still keep as the live tissue, but here in this kind of a model, you have the presence of all the blood flow and all the signals that comes via the bloodstream, hormones, etc. Now, this animal we put up on the microscope stage with the kidney into a little dish. And so we now can image this on the microscope because we are injecting different dyes. And that is shown to the right. Now, in red, you see the rhodamine, which is a rhodamine conjugated dextran, which is large enough, so it's not filtered through the glomerulus. And that is why the red outlines the blood vessels. In blue, we have added Höxt in this case to reveal the nuclei of the cells. And in yellow-green, you see the autofluorescence of the proximal tubule cells. And those, for instance, here is one proximal tubule, so here are the cells. So this is the lumen where the primary urine is flowing. And in between here, you see the, in real time, it looks like this. So that now the black streaks that you see moving, that's the red blood cells that are moving through the system. Using this kind of fluorescent techniques also, of course, require then that the bacteria can be detected. And so to do so, we made a construct where we base this on a clinical isolate. It's the CFT073, which is commonly used bacterium in the pyelonephritogenic research community. Now, we found that if we inserted the GFP under the control of the TET promoter in the absence of the repressor, this construct is inserted onto the chromosome in a site which is known to be inactive in the E. coli system. And we found that indeed with this construct, we have very nice expression if you compare the GFP in the in vitro situation as well as in the in vivo situation. And I just want to emphasize that it's quite common that people use the RPSM promoter, and that one is completely useless because it regulates ribosomal proteins. And when bacteria adapt to the growth conditions, of course, ribosomes, that's among the first things they will regulate and downregulate. So that one is crappy, and the PTAT worked nicely. So now, how do you obtain the urinary tract infection and pyelonephritis? Well, in the usual case, bacteria will enter through the system via the bladder, ascend to the kidney, where they now can reach into the tubular systems. Thing is, though, that if you elicit this infection in a rat and then go and look at the tissue 4 days later, you will have all possible kind of structures in the tissue. Some areas which are completely uninfected, and that is going to be the majority of the areas, while others have severe tissue destruction, or you can see some areas where you have bacteria present at the very early time frame, where you still maintain very nice tissue morphology. And I'd like to mention that these are confocal images from fixed tissue sections in this case. So what this tells us is we need a system that allows spatial and temporal control of the delivery of the bacteria, because we want to know when did the infection start. And to do so, we are using a microinfusion system, so that when we have the kidney exteriorized like this, we can use these very sharp needles, which we inject then 10 to the 5th bacteria in a volume which is between 0.1 to 0.7 microliters. And we do this very slowly, so that we follow the same rate as which the primary urine is filtered and going through the tubules. Now, you notice that we have two capillaries here, and that is because the left one, we are injecting the bacteria, and you see that mixed with the bacteria, we have added a blue dye so that we can identify the injected tubule, because we expected that at an early time frame, we will have very few bacteria present. Now, the other one is injecting some oil dots. So here we see 2 oil dots, and those we use in order to orient us when we take the rat from this stage onto the multiphoton stage. And so then looking at this infection, it looks like the following. Already an hour after injection of bacteria, you can see a few green bacteria that has adhered to the epithelial lining. And the blue here is the dye, which is endocytosed by the proximal tubular cells. So we know that this tubule, this is the same tubule, it's just that it is winding through the tissue like this. So we have cut twice through this. We're looking at 2 sections of it. And those bacteria, you can see in the bottom image that they are nicely attached to kind of this tunnel opening. But already after a few hours, you see the massive bacterial multiplication. And the tubules are completely filled with bacteria. Now at this stage, this was actually our first experiments. We put the kidney back into the peritoneal cavity, and we incubated the rat, or let the rat We kept the rat overnight, and then the following day we could take out the kidney and again image exactly the same site. And as you see, we have a dramatic tissue destruction. But interesting, the bacteria are gone. Now, at this stage, we dissected out the kidney and looked for, using the confocal microscope, what is actually present in this dark area. And as you can see, we have a huge infiltration now of neutrophils, and in a few of them, we could identify green bacteria that were now all ingested in the neutrophils, but no extracellular bacteria. Also, what this image shows is the very localized nature of this response, because to the right, that is if we now, at this stage, when we looked at this image, if we move the microscope just 2 fields to the right, you see that we have absolutely normal tissue morphology. Now, that experiment told us that really 22 hours is too late, then it's all over. So we wanted to look at the earlier time frame, to actually look at the kinetics. How do we reach this end result? And so we have kind of standardized our studies to go from 0 up to 6 to 8 hours. And what was striking in our first series of experiments was that you notice that the red blood flow is completely abolished within a few hours. And so this was accompanied by severe consequences from the vascular perspective. And that is that, first of all, it seemed like we have like congested vessels, so that we got these big, dark silhouettes that represents cells present in the bloodstream coming to the site. We got the congested vessels. This indicates plasma streaming. That means that it's only the plasma that is passing and that contains the red dye and no red blood cells. And also in the image to the right, you see that now we have also perivascular leakage from the vessels. So apparently this is quite disadvantageous from the vessel's perspective. So we want to determine now the rate, how quickly do the red blood cells move in the vessels at the infected and non-infected organ. And so you see here we have used the UPEC, the wild-type bacterium, and we use the PBS injection as our control. And what you notice now, if we can maybe make it a bit darker, I hope we can see on the, maybe we cannot. So on the bottom panel, in this panel, we have the blood vessels where we can see the black streaks that represent the red blood cells. And now if we look at those, in those two, you would see that they are absolutely horizontal like this, while in the infected one, we can see that those ones, we have a slope on those dark streaks that represents the red blood cells. And that means that the multiphoton microscope now catches more images of this, of one red blood cell as it's moving, and that is why you get the tilted angle of it. And simply by using the ordinary equation for the rate, we can then plot that in the in PBS injected one, the rate of the red blood cells stays normal, while in the infected one, we rapidly drop to 0. This goes actually to 0 millimeter mercury. No, eventually, I will come to that in next, sorry. I'll get that in a second. That is what comes here. Now, the fact that we dramatically reduced the speed of the red blood cells in the tissue, that prompted us to now go and actually probe the oxygen tension within the tissue. And so to this end, we turn to experts in this area, which now can use, where we put the rat is down here, and it takes a huge lot of equipment to do these experiments. And it's based on Clark-type electrodes that we insert into the injected tubule right after we have delivered the bacteria. And the result from these experiments is shown here. To the left, we have the data from one animal. And so you see in the one site, we inject the PBS and we monitor that site, while in the other one, we inject our bacteria. So now we have the infection and the control in the same animal. And you notice that in the PBS infected one, or PBS injected one, the oxygen tension stays normal, while in the UPEC infected one, we get a rapid decrease, actually down to 0 millimeter mercury, while in the PBS injected, it stays at around 48 millimeter mercury. Now, this is very local response because we also did measure the arterial blood pressure, and that one showed no changes over time. So this experiment we performed in 7 animals, and you see that in the graph to the right. And now you see the striped bars, that's naive cortex, and the black ones, that's the PBS injected. And again, the gray ones that represent the UPEC shows that this dramatically decreased down to 0. So, now that the oxygen disappears, why is that? Well, one hypothesis is, of course, that we have an increased consumption of oxygen at the site of infection. And so this slide shows that the vessels, the peritubular vessels, those are the ones where the blood disappears, whereas in the glomerulus, this is the glomerular structure, here we still maintain the blood flow over a longer time. So that tells us that we have some very specific signaling events going on from where the bacteria are located across the epithelium that affects the very local vessels out here. And so to analyze whether we had an increased oxygen consumption, we prepared primary preparations of proximal tubular cells, and we performed a in vitro experiment where we record the oxygen consumption, either in the proximal tubular cells themselves, where we look over time at 50 minutes here. And you can see that compared to this, if we use the bacteria per se, they do not cause any increase in oxygen consumption, while if we stimulate these cells with our UPEC bacteria, that causes a significant increase in oxygen consumption. And that in turn may be due to the increased metabolism in these cells. And so we recorded some of the pro-inflammatory cytokines that are shown in the graph to the right, where we show for the proximal tubular cells as well as from the renal tissue. And you can notice that the TNF-alpha, IL-1 beta are both upregulated in both situations. Now, this is at a 50-minute recording after infection, and that is too short time to recognize IL-6, but if we wait for longer, also IL-6 will appear in the PTC preparation. Now, reducing the oxygen in the area will have severe consequences. Now, it's well established that when you elicit an ischemic, or when you have an ischemic situation in the tissue, you will have severe consequences for the cell adhesion and viability. And that includes that the cells undergo necrosis, apoptosis, after they have lost polarity, and then are sloughed out into the lumen, the tubular lumen. So we then imaged one tubule over time, which is shown here, where the same tubule is imaged from 2 to almost 8 hours. And so you notice that now bacteria start to multiply, and with time, all these round dark areas, which contains lots of blue, appears in the lumen. Now, these are clusters of epithelial cells that have been sloughed up from the upstream epithelium and now enters into the tubular lumen, and that's where they are now floating down together with the bacteria down through the lumen. Another thing to note is that with time, you see that the bacteria ends up on the wrong side, so to say, of the blue staining, which means that these cells, the epithelium, has lost its polarity. So if you consider actin rearrangement in ordinary ischemia, and ordinary in this case, that's from the renal researchers' perspective, that is when they use a pedicle clamp to elicit ischemia after 25 minutes, and then they look for actin staining. And you can see that as compared to the control, in the ischemic animal, you will have all this blebbing of the actin of the cells and reorganization of actin staining. And notice how well that mimics the infected tissue. Now, to the right, you see those two, that's the tubule that is infected, and these ones are also proximal tubules, but the non-infected. And you can see a dramatic difference in the actin staining. Now, what this also showed is that you see here that the bacteria seems to start to penetrate between the cells. And that is actually something that we observe very, very frequently. And not only do they penetrate between the cells, but also they are kind of sneaking in underneath the epithelium. So now they seems to line up against the basement membrane. And apparently that basement membrane turned out to act as a second barrier to for the dissemination of bacteria. Because here we have stain now with the collagen 4, and that is a major component of the basement membrane in the proximal tubule. And so you see that now in this area, bacteria are nicely lining up against this, but eventually with time, they will also pass this barrier. A very interesting thing here is that these kind of signals that are transmitted across the epithelium appear at an early time frame before bacteria actually start to penetrate across these barriers. Because remember, the ischemic response was elicited already after 3 hours. And that is actually before neutrophils are recruited to the site. And that tells us that there's a massive signaling cascade going on at this epithelial border where bacteria are recognized and that recruit the proper cells, but also elicit the proper responses in the tissue. And so adding movies to a submission nowadays, that doesn't work if you only have images. You need the molecular data. And so we were quite happy that as we know exactly where the infection site is, we can dissect out that very tiny piece of tissue and now do ordinary molecular analysis of RNA, for instance. And this is just a PCR showing that we indeed can identify the different markers for the infected tissue versus the uninfected tissue. And we have used this now in array analysis in order to look for the host genes that are initiated during the infection process. And one of the things that came up was that several of the genes that are involved in the clotting cascade are upregulated. And when we realized that, we went back to our images because suddenly we could start to explain these black areas in the vasculature where it was dark cells that were too small to be red blood cells or other cell types. And so our guess was that it was platelets. And indeed, when we also found that several of these genes involved in the clotting cascade are upregulated in the 8-hour infection, that led us to the conclusion that it seems like clotting is induced in this tissue. So of course we wanted to know what's the reason for the clotting. Why would that be? So in order to abrogate clotting from occurring, we injected the animals with heparin. So we first infect them and do the surgery, and then we inject heparin. And you can see in this top graph that indeed the blood flow stays longer as compared to in the previous situation, although eventually it reached the same end result. But during this time frame, all rats died. Between 6 and 8 hours, they all died. And this was accompanied by in their blood pressure. And that is, of course, connected to the sepsis, we thought. And so, of course, then we harvested the blood as well as the different organs, liver, spleen, and heart. And in the normal situation, we would only see bacteria present in the urine. We have never found bacteria inside the animal. No other animal has ever died on us in these experiments. But now with the heparin, suddenly we have bacteria everywhere in the animal. So this really tells us that the ischemic response is of huge importance in order to encapsulate bacteria, to keep them at the site, so that you give neutrophils the time it takes to come and clear out the infection. So in this slide, we just compare ischemia with the reperfusion in the normal situation, where you have the loss of cell polarity, blubbing, cells are sloughing off, but eventually you reach the stage where you now will have the spreading of cells and dedifferentiation, and eventually you get back to the normal situation. Now, that doesn't happen with our bacteria. Here, you will start to destroy the epithelium when the ischemia is occurring, and eventually this will now encapsulate, keep the bacteria at the site so they do not spread into the systemic circulation, and that gives time for the neutrophils to engulf these bacteria. So we conclude that the bacteria, first of all, they signal from the outside in the lumen across the epithelium. That's where the signals are initiated that leads to the ischemia in the tissue. Secondly, that this response encapsulates bacteria, and you see on the left-hand side, this is after 24 hours, and you see that the site is extremely well localized. And if you look to the right, that shows that the blood flow is working perfectly just to the border where this big foci is located. So we actually believe that the ischemia is, can be added to the innate immune defense mechanism towards these bacterial infections. So, so that now brings, this slide I just showed you recently that we can do the molecular data. And from our perspective, knowing now that the host has such thing happens in the host, of course that has to matter for the bacteria. If you consider bacteria that are injected at time 0 and the environment they meet, as compared to 3 hours later, when you have depleted the area of oxygen, of course the microbes will be experiencing a different microenvironment. And so in this case, we started off by looking for whether the bacteria express alpha-hemolysin. And again, we can do this PCR analysis because we have a very localized infection, so we know where bacteria are located. And so they are not, the mRNA is not diluted by all this uninfected tissue. And indeed, we can see, using the 16S RNA as control, that the alpha-hemolysin is expressed in the infected kidney, but absent in the non-infected. And so we then wanted to look for the effect of this toxin. And this toxin is expressed by this uropathogenic E. coli strain in a bit more than 50% of them. And it is considered a virulence factor for UTI infections. And what you see here is the wild type on the top panel. And if you compare now the wild type, or the UPEC strain, which has the hemolysin mutated, you see that the blood flow stays for longer in these animals. Now, this we haven't done the in vivo calcium imaging yet. We wish to do so. But at least this corresponds very nicely to our previous data, which shows that this toxin, alpha-hemolysin, elicits calcium signals in the proximal tubular cells in an oscillatory manner, so that the calcium oscillates in the target cell. And it's quite established that the frequency of calcium oscillations, that is what dictates the physiological outcome in the tissue or in the cell. In our case, the hemolysin induced an oscillation with a periodicity in the 12-minute range, and we could show previously that that leads to the production of IL-6 and IL-8. And so we believe that knocking out alpha-hemolysin in this situation, that means that we do not get that early IL-6, IL-8 response, and that may be part of why we see a delayed response in the tissue. So what we hope now is to see that this journal should kind of shift its into the next generation of journals, molecular microbiology, cellular microbiology, and now we hope we will start to see the tissue microbiology journal, because we think that this interaction, which is so dynamic between how the microbe experience, what they do to the host, how the host then affects the microbe, that balance is going to only be possible to study in this kind of animal model systems. So, of course, this has now huge implication for what we are doing currently, and one of the major things is, of course, that knowing that we have this ischemic response, we are now a lot interested in how the obstruction of the tubule will affect the infection rate, but also and the glomerular flow rates. And this brings us into of course, the role of bacterial virulence factors and how do bacteria adhere to epithelial linings and get up through the system. So it's the interplay here which is of main importance. Now, the thing is, though, that doing this in the animal model is a bit tedious. So we are thinking of other ways that we can do things, and here comes one of those that I think Philippe introduced this, saying that Collège de France shall not bring established knowledge to the audience, but rather the idea of free research. And this is that kind of free research, I think, that we don't know yet if this will work. But the idea is we want to create a model system, in vitro system, where you can utilize several different cell types in a very well located manner on the surface, so that you can add your bacteria over here and study this cross-talk between cell types in an in vitro assay. And to do so, we have now utilized, together with the partners at Linköping University, that's headed by Professor Magnus Berggren. They are expert on conducting polymers. And conducting polymers, that is, I have depicted them up to the left. We use the P-dot polymer, which is doped with the tosylate. And together, this it becomes doped, and so that gives very high conductivity. In this experiment, we have now made a gradient so that this material, when you apply a potential to this material, it will become reduced on one side and oxidized on the other. And here we have a specially designed device so that now this area in the center, that is where we establish a gradient that includes all redox situation from left to right of the material. And now we have put a ring on here, which is like where we can put our cells in cell culture medium. And we can now start to grow cells on this surface and see how where they interact with the surface. In this case now, we can vary the source, the drain, and the gate potentials. And by doing so, you see that the cells, and these are the MDCK renal epithelial cells, you see that we can play with where on the area. So this from left, position 1, to right, position 7, is then corresponding to the left to right in this gradient. You see we can direct where cells are going. So if we now can start to modulate where this cell type is going and then add on another and another, we hope that we can create a device for studying intracellular communication in vitro. These materials are printable, so they are very easy to print on support. So this is stuff that we are doing then in the context of the Swedish Medical Nanoscience Center that I hope you will hear more about in the future. And eventually the acknowledgement for my team members that have been crucial for this work. It's primarily Lisa Månsson who initiated all the work on the multiphoton microscopy. She's now a postdoc in Vancouver. And the other part of the infection model has primarily been performed by Kira Melican and Jorrit Bokel, while the others are more involved in the organic bioelectronics. So thank you for your attention.
Thank you very much, Agneta. It was splendid. For those of you who wouldn't know, Agneta got a PhD in civil engineering, is that right? And so you can see the engineering.
I'm a chemist.
Huh?
I'm a
chemist. I thought you had some training in engineering as well.
I'm a chemist.
Dreaming?
Yes.
Anyway, so are there any questions?
So I think in the first part of your talk, your data would be also consistent that the oxygen consumption comes from the production of radical oxygen species, which should be also consistent with the initiation of innate immune response. Have you measured that as what is its contribution as opposed to ischemia?
No, we are looking a lot for different systems how to do that. And indeed, we think that that may be of crucial importance.
Can I follow up on this question? You are putting up this model of encapsulation of microbes by platelets and so on. So this is, I have nothing against this model, but could it be that we have actually underestimated the role of ischemia in inducing inflammation? Did you, for instance, check TNF level after heparin treatment and see if TNF goes down, in which case that would be part of the spreading process?
We haven't tried that, but indeed I agree completely that the ischemia itself is known to have severe consequences for inflammatory reactions. That goes the same with the obstruction. Right. And that is my point, that we need to merge these fields so that we are not looking at independent events, but rather the combined effect.
Because in a way what kills in myocardial infarction is more inflammation than the ischemia itself.